Martin, welcome to Kiel Life Sciences, KLS. You are Senior Science and Technology Advisor with 10x Genomics. You are fascinated by differential gene expression all your life, as you told us. You got a PhD at the Technical University in Munich, contributed enormously to human genome, human epigenome, differential gene expression, and has key papers in Science in Cell, changed the system and went from a university system into a non-university, more industrial system, founded your own companies, and are now at 10x Genomics. And still, there seems to be a red thread throughout the whole career, and you are still doing kind of what you probably were thinking of in the early days. So an interesting question always is to ask, how did all that start? Where did you get your interest? Why molecular biology and why differential gene expression? Was there key moments in the teacher, or how did you get to where you are right now?
Well, actually, thank you very much, first of all. It's a pleasure to be here. Where did it all start? Well, actually, it started that I wanted to be a physician in first place. And then I probably was not courageous enough to be it. I wanted to have a job where I can do mistakes in a way that no patient suffers from it, but still being interested in all the mechanisms of all human beings, but also from all, well, in life in general. How does it occur? Mm-hmm. And a lot of that really goes back to the early days, looking into the environment. Well, I grew up on the countryside and was always fascinated from nature. And I always wanted to know how this all works together. And later on then in my studies at university, it occurred to me that it all, of course, comes down to genomic information, but what is made out of that? And what is made out of that, in first place, is transcriptomics and is gene expression and how this is regulated. Mm-hmm. So that's how it started for me. And that went on from the very beginning with methods which are hard to explain to young scientists today. Over different incarnations of technology, now directly into the newest format that we look not only which genes are expressed, but also where they are expressed, mm-hmm. and look into the tissue directly and try to better understand what it keeps all together and how it evolves.
Very interesting. We talk a bit later probably about it, but starting as the wish to be a doctor or a physician, and your research and your activities are still very, very human-oriented and patient-oriented. We can talk about that later. But maybe coming back what you said at the end, can you summarize us and the audience in a few sentences? Where is the research you are doing now? We have invited you. You gave yesterday the opening keynote talk in a summer school called Spatial Biology. And that's what you just alluded to. We are entering a new world by realizing that life processes also have a spatial and a temporal dimension. And this is what you are doing now and already for a couple of years. Can you summarize us a bit how these efforts are to get spatial information, and what is the current research which you are supervising and guiding at 10x Genomics at the moment?
So far, we were always looking into gene expression and gene regulation processes in a way that we took out of an organism some tissue, we minced it, we looked into that by methods of sequencing, by methods of transcriptomics, and we tried to better understand which genes are expressed and try to better understand, for example, as well, which genes are causative for certain disease and disease processes and so on. But it's always a mixture, and organisms consist of many cells, billions of cells. That is well, even trillions of cells, probably, depending on the organism, and they are all organized in tissues. And this organization is per se an informative layer. Where are things happening in a tissue? And what is now possible with the new methods, it is just looking into the cells, but in the tissue, you don't have to mince the tissue anymore. You still need to cut it at the moment. You need to have tissue slices, like it was done in pathology for many years, but then you can directly also add these molecular layers in this, on these tissues which are on a glass slide, and you can see where genes are expressed.
So what can you do now? What can you see now what we couldn't see 10 years before? You are now at a cellular level in different organs. Mm-hmm. And what can you see, and how make you these molecules visible?
Well, from our perspective at 10x Genomics, we have two technologies to make this visible. One is actually a way where we put the tissue on a specific slide where we can do afterwards a single cell dimension resolution by pulling out nucleic acids out of the cells. And in principle, what we did in bulk, do it on a spatial trajectory on spots which are about 2 micron in size. And we get a whole transcriptome on these little spots, and we better understand which cell types are there. We better understand which cell types are neighboring each other. So we also get an information, for example, in the tumor tissue, where the tumor is sitting, tumor cells are sitting, and where the immune cells are also located within this specific tissue slice. And the second technology is then different. We no longer try to pull out anything out of the cells, but we bring the detection technology directly into the cells. So the surface is in principle, again, a glass slide, like a microscopy slide. And it's in principle something which is a molecular microscope, where we bring in the detection technology for transcripts directly into the cells, and we can measure, even in a subcellular manner, directly where genes are expressed. And in addition, we keep the microscopy image for both technologies, by the way. So that is what was done for many years, staining the tissue, seeing where cells are located, seeing the cell borders, seeing the nuclei. But what we add is, in principle, the molecular layer on top of that. So what we before only had in bulk sequencing, the whole depth of transcriptome analysis, we can now do directly within the tissue. And that's the major difference.
Important is that you not only rely on candidate genes, which you know already beforehand that they are maybe tumor-promoting activity, but is it correct that this technology also can be applied to just the whole active genes without knowing what they are really doing in terms of, I bring the keyword, novel genes or orphan genes or mutated genes in a given tumor?
Yeah, actually, this is a slight difference between the two technologies. Visium allows to do that as long as the genes are, or the transcripts are polyadenylated. We can capture now, in principle, all transcripts. And as it is sequencing based, all what is captured can be sequenced. So we can capture those genes which are known, but we can also capture those which are unknown, as long as they are polyadenylated. We have a second modality where we do this with a probe-based approach on Visium. There we normally have whole transcriptome analysis for human and mouse, but for the known genes and basically mainly the protein coding genes. And with Xenium, we have the higher resolution still, because we can really do subcellular positioning, but we are relying on panels which are pre-designed, which are in the version 1, around 400, and with a different modality up to 5,000. So that's the prime modality. And you can add genes and transcripts on a customized basis. But that is going not into the unknown at the moment, because you need to have pre-knowledge to be able to design the probe sets before. But with Visium 3', you can definitely also look for yet unknown ones. And you are species agnostic. on that.
Yeah, yeah. Coming back a bit more general to the beginning and your desire to do something good for patients or for humans, I mean, your technology and technologies in the spatial biology field, which other people are using now or developing now, it seems to me that the patients get more and more transparent. This is enormous, how deep we now can look into what happens into this sick tissue. Nevertheless, there is an ever-increasing number of cancer patients. It's actually going exponentially, and there is no way that we can somehow get control of that. For me, that's a discrepancy. We learn more and more about what's really going on if you compare a healthy organ versus a sick organ. But in general, can we ever help a patient to get healthy again?
Well, the mission 10x Genomics is on is really to make disease curable. And this is indeed something where we strive to help to achieve this mission. All what we do at the moment is research purpose only. I just need to say that and state that as well. It is for clinical research and so on, but it has not reached the status that it would be directly applicable in routine diagnostic or something like that. Nevertheless, the strong belief is that the better we understand the mechanisms behind the disease, the better we understand the mechanism behind cancer, for example, the better we understand the crosstalk between cancer and the immune system, the more we can also understand what kind of treatment is efficacious, the more we can really also probably treat patient according to their individual needs. And there are a lot of treatment options out there which are sometimes extremely efficient, like immune checkpoint therapies, but not to all patients in the same way. So the responder, non-responder situation is clear. The responders have, in part, tremendous improvement of their health conditions. And the non-responders, of course, is more difficult because they have side effects from the treatment. If we only can really decide who will be a responder and a non-responder, it would be also already a major achievement for the patient. On the other hand, with the spatial analysis, we can now really see where the immune system is sitting in a tumor. And this is not well, even if the tumor species, let's say breast cancer, is the same, it looks quite different on the individual basis. Where immune cells, which immune cells are sitting where in the tumor, and what they are doing there still needs to be better understood. But in a way, we have the strong belief that, of course, the better we know that, the more we can also develop targeted interventions for that. Or probably not we, because we are a technology provider, but our customers, the researchers out there, can really use that to better treat the patients at the end of the day. But at the moment, it's still research-based only.
I thank you very much. I think this is a very valuable comment. I miss one word still, and so I come back now. What Key Life Sciences is actually, the mission of Key Life Sciences is evolving health in a sustainable environment. And what we are in many different levels, in many different organisms, from the clinic to the plant sciences, to the invertebrates, to the vertebrates, are doing, trying to understand how does the environment influence the gene expression and the fitness of the organism, and that under an evolutionary time scale. And so coming back to this ever-increasing number of patients which are suffer from diseases, some of them are very modern diseases and were not really known 100 years ago. Isn't the key component the changing environment where we are living? And the environment in this respect I define very wide. Can be outside, the physical environment can be anything, but can be also the immediate environment of the organs and the intestine, which is a microbial world, which you cannot separate anymore from the host tissue, and which was with us all our evolutionary time. So, getting back to your key statement of the variability, and there are responders and there are non-responders, wouldn't it be interesting to think about that these differences is maybe due to the microenvironment of the two patients? One is a responder and the other one is not a responder. And we have evidence to believe that because the success of chemotherapy is associated with differences in the microbial world.
You're absolutely right. And one environment definitely is, for example, nutrition. Yeah, this is something.
Key point.
A key point.
Yeah.
This is what we eat.
Yeah.
And how we eat it, and how it was produced, and so on. That is, I think this is out of question that this plays a role for health status, and probably, I mean, the rate of diabetes is heavily increasing in all Western countries and beyond. Sugar intake is creating issues. Sugar intake is creating probably also inflammatory status aspects, which are directly also influencing other disease, maybe even cancer and stuff like that. And of course, you're right. The environment plays a major role in that. But on the other hand, if we better on the molecular level can then also see whether two patients coming from different environments are also different on their molecular level, the better probably we can also take this into account and probably also build classifiers taking those things directly into account. But you are absolutely right. The other point is probably physical fitness also in the format of workout and stuff like that. Sports, we are all sitting way too much. We haven't understood how this is influencing us, but nutrition is something which is very clear.
I think everybody agrees to that, and for me as an evolutionary developmental biologist trying to understand basic principles of life, it's very clear that we started with a very simple model because we thought this is easy to understand, but it's getting much more complex the more we know. And so now comes the tissue complexity, the epigenomic complexity, the microenvironmental complexity. And so the more we dig, the more complex it gets, and solid answers and solutions will still have to wait. Yeah.
Absolutely. And you just mentioned efficacy of chemotherapy before. I think a couple of years ago, I think it was 2018, there was a nice Nature title, Bacterial Snack Attack, where it was just shown that, I think it was pancreatic cancer, that bacteria can metabolize certain chemotherapeutic compounds. Sure. And that is having also direct treatment effect, if they are there or not there.
Where will the field of spatial biology be in 5 years?
Very interesting question. I think, well, first of all, and we have discussed that as well, probably it will be closer to the patient already. Probably still not in routine, but in clinical trials and so on, so in clinical research. We will see more. There are additional modalities which are just being developed for spatial technologies on the protein level, on the metabolomics level, etc. And I think also a multimodality aspect will come to play in spatial biology. That is something I'm strongly convinced that we need additional modalities to really better understand the whole complexity in disease and in living organisms in general.
You are a wonderful example that when you see how science goes, there is not only one way of how to proceed with your career. And so you left the university system at some point, but you continued actually what drives you, and you are very successful, and you're now a senior advisor at a major global company. What made you do this decision? I leave now the classical, canonical, standard system, and I'm tough enough now to look for other things. And many young students and early career scientists are having difficulties when they see the system and how rigid it is, the university system, and don't see really that there is a wide possibilities of making their career. What made you leaving the system?
Yeah, I wanted to see how the world outside the academic system looks like, actually. Curiosity on that level. And on the other hand, it was, in principle, and first transition was a really smooth one because I was a customer of the company who then, in a way, pulled me over to the commercial world. And I have to also admit that the CEO of this company at the time had a very, and still has, had a very inspiring approach to combine the curiosity from academic research with commercial success. And that made me interested in that. And of course, also from a technology perspective, the company could do things and they had developed databases, technology, and so on, which I found extremely interesting. Nevertheless, when I shifted to the commercial world, I thought my scientific life would be over, more or less. But finally, it wasn't. Because I realized that in this still startup environment, we were around 20, 25 people at the time, we could involve the company in many research projects as industry partner, as technology providing partner, and so on. And that allowed me to continue also with science. So leaving the one was not in conjunction with completely stopping my scientific interests, let's say it this way. So this is possible, probably not all the time, but in this case it was.
It also depends, for sure, it depends on the personality.
Yeah.
If an early career scientist comes to you now at lunch and asks you that she or he is in a kind of a decision-making process, maybe leaving science at all, or not sure if one can make it, and one sees all these glossy models and glossy papers and glossy PIs which have made it, dear Dr. Seifert, what does it need to have success and to make the career? What would you answer?
Follow your motivation, have fun, and follow what you really want to do. That is basically what I would give as an advice, and don't be afraid. If the graduate students, and I had a conversation with one yesterday, and she asked me, what should I be afraid? And said, no, you're all trained so well that also in this difficult world we are in, you will always find a place. And in many cases, it is also not, and I can just say that it's not plannable in a way. If I wouldn't have done gene expression analysis in zebrafish and would have needed transcription factor analysis technology, which I could only have from this one company, got into contact and so on, I wouldn't have landed there. So sometimes it is just also happening by chance to a certain extent. But if there is a chance and your gut feeling tells you that could be interesting, and I could, at least for the next couple of years, probably like what I can do there. And if I don't like it, I still can make a decision which then probably leads me to a position which I like more. Don't be afraid, just do it.
That's a wonderful sentence at the end. I think don't be afraid, follow your gut feeling.
Yeah.
We know that the gut is full of microbes. And so they may influence you.
And sometimes the gut knows more than the brain.
For example. We are very happy to have you here in our summer school. Thank you so much for coming, and all the best wishes for your future.
Yeah, thank you for having me. Thanks for the invitation. It's a pleasure to be here.
Thank you so much.